Hello everyone and welcome to today's webinar, Catapult Your Cell Therapy Research into the Clinic, CAR T-Cell. I am Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speakers will answer questions via email to the addresses you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Dr. Annika Graband, expert for flow cytometry reagents, Miltenyi Biotec. Dr. Melanie Rietenbach, expert for engineered cells, Miltenyi Biotec, and Dr. Saskia Rösch, expert for T-cell immunology, Miltenyi Biotec. Speakers, you may now begin your presentation.
A very warm welcome also from my side to our Translational Cell and Gene Therapy webinar series. And please acknowledge also our disclaimer here on this slide. And the agenda for today for the first part of this webinar series. First of all, we would like to introduce the path to CAR T-cell therapies and have a look at your mission and at your vision and our mission. Then we will dig into the translational research workflow, followed by the clinical manufacturing platform, the Prodigy, and what's new there, what you can do with it. And then we also will have a summary and point out some helpful resources. So I would like to start off with a question today about your vision when it comes to CAR T-cell therapy. I'm pretty sure that most of you have a very clear vision to move your CAR T into the clinic and successfully treat cancer patients. And during this mini webinar series, we would like to highlight how Miltenyi can support this very meaningful task. So as we all know, the path to CAR T-cell therapies is quite a long one, starting from basic research and discovery, including target identification and validation, as well as optimization and lead identification. So the developmental phase, so the preclinical development, to several clinical phases, to a finally approved and marketed therapy. And pursuing this vision requires a lot of efforts, and you need to overcome challenges, and it goes along with a very steep learning curve. However, this is doable, and during the last decades, the field gained a lot of knowledge, and by today, already 6 CAR T-cell therapies are approved. More than 1,150 CAR T-cell clinical trials are ongoing, and over 140 companies all over the globe have CAR T or TCR T-cell therapies in their preclinical pipelines. So every phase comes along with different needs, and it requires different and very tailored solution. And as Miltenyi is very active in the field of cell and gene therapy, especially when it comes to CAR T-cell therapy, from early research to large-scale clinical manufacturing in our cell factories, we exactly know about the hurdles, as we are somehow our own customers, so to say, and we do save the same problems. And therefore, we develop and produce tailored solutions that we are using in-house, but we also market them. And today we will focus on the translational research workflow as well as on the clinical workflow solutions. So this is basically the workflow we are following, both in small-scale research setting as well as in the large-scale clinical manufacturing, applying the TCT process. And today I will focus only on cells, on the cell set part, due to the sake of time. So from human blood products to T cells, there are different options published and are used. So for example, positive isolation of CD4 and CD8 T cells, or untouched isolation using the pan T cell isolation kits, and some culture simply PBMCs with cytokines and dedicated media. And if you are in a research setting, this is all fine with pros and cons, but especially when it comes to when you envision to translate your workflow from small scale to the clinics to a large scale manufacturing, there are some points that you should consider. So, for example, are my results really reproducible? Are my reagents available in GMP quality? Is the isolation method that I am using and establishing in my research setting also translatable to an automated system? And is the clinical process already available? Is it established? Is it programmed on a automated system? And yeah, to meet all these challenges, so we in-house go for CD4, CD8 T-cell isolation. And yeah, here you have basically in a research setting 2 different options. The first one is via the PBMC isolation step via a FICOL. And then you can use a 1-on-1 mixture of the CD4, CD8 microbeads. Or the second option is to go directly from blood products, skip the FICOL, and yeah, in only 30 minutes, you will get your T cells from blood products to CD4, CD8 T cells using the straight from CD4, CD8 microbead kits. And taking a look at the comparison data of the FICOL approach versus the straight from approach, you clearly see, I don't want to go into detail in this webinar series, what is clear, what becomes clear is that no matter what approach you decide for in terms of T cell activation, transduction efficiency, as well as regarding the function of your CAR T cells, there's no significant differences between the 2 different methods. So they are really comparable. And what I also want to highlight is that CD4, CD8 isolation, there is a very low variability between the donors that also underlines the reproducibility. And now I would like to show you why we recommend the CD4, CD8 T cell enrichment at the beginning of our workflow. So by the CD4, CD8 selection, you will start your process with a very well-defined enriched population of T cells, which is key for specific expansion and also higher reproducibility. And yeah, taking a quick look at the data here. So we analyzed 3 different donors. We started with Buffy codes. And by the CD4, CD8 positive selection, we increased the purity by reducing contaminating cells, such as they are listed here, such as granulocytes, B cells, or monocytes. And by this, we also got enough T cells to ensure a robust and reproducible T cell activation and expansion. Yeah, so to sum up this part, so CD4, CD8 T cell isolation using the max microbeads, then you will start off with a well-defined and rich population, which provides a higher reproducibility for your downstream process. The CD4, CD8 microbeads are available in max GMP quality for an easier translation. And the positive T cell isolation is also compatible with our Clinimax Prodigy TCT as well as TCE process. And the max microbeads, they are already used in clinical CAR T cell trials worldwide. So yeah, this was part 1 of the whole workflow, so the cell separation step. And what I also would like to highlight here is that we have some free resources available. So for example, if you scan the QR code here, you will be directed to our R&D research protocol. So this is basically the small scale TCT protocol to engineer CAR T cells. Then we also have a very nice application page summarizing all the solutions and product highlights, as well as scientific posters, and our brand new CAR T-cell research and discovery brochure. So with this, I am at the end of my talk. I thank you from my side already for listening, and I will hand over to my colleague, Melanie. Thank you.
I would like to continue with our clinical part. So we have the Clinimax Prodigy platform, which is supposed to tackle most common challenges in the manufacturing part. So some of you might be aware, some not. So with the Clinimax Prodigy, you can really do the whole workflow fully automated and standardized, really from cell processing, selection, activation, gene engineering, expansion, and then also the final formulation and fill. And it's still flexible enough to adapt a lot of processes that you might have to the system. It reduces your costs because it's really fully automated and a closed system. It minimizes your time to market and delivers a consistent product quality since it is an automated system. And just recently, last year, we added 2 modules to complete the platform. So now we have the CliniMax electroporator available as well, which will now integrate cell electroporation into the full workflow. And we also have now the CliniMax formulation unit available that now offers the possibility of final cell formulation and final fill to complete the whole workflow. And when we think about our T-cell manufacturing options, we already have quite a few to offer to you. So our most widely used one is our T-cell transduction system. It was already released in 2016, and it's really widely and globally used in clinical trials. Then we are working on a T-cell transduction large scale. It's currently under development, coming hopefully soon. And what we released last year together with the electroporator is our T-cell engineering process that now offers also the possibility for cell electroporation in a closed fashion. And last but not least, we offer a customized application service to customize your process on the device if we don't have an off-the-shelf solution, and that's on demand. So let's start with our famous T-cell transduction system. So our CliniMax Prodigy T-cell transduction system, this process offers you the possibility to select your T-cells from your heterogeneous starting material, for example, whole blood or buffy coat or leukapheresis. Then the T-cells are activated, they are transduced and expanded, and then can be harvested afterwards. And the good news is that all the consumables that you need for the whole process, you can all get from us. So it's one purchase. And we have already a lot of experience with that process. So it was already launched in 2016. And since then, we have built 2 manufacturing sites where we are manufacturing for our clinical trials. And we use this application there as well. So we've already treated our first patient in 2018. And since then, it has been ongoing. Like I said, last year, we extended the platform with the 2 new modules, the CliniMax electroporator and CliniMax formulation unit. And of course, our customers are also using this platform quite widely. So this is a track record of publications with our T-cell transduction system. And what you can see is that there were a lot of publications in the last recent years about how to optimize the process and how to translate the process onto the platform. And now in orange, what you can already see are first publications of clinical trials. And of course, a lot of clinical trials are not disclosed, but there are a few that I can share with you. And those that you can see here, they're all using our T-cell transduction system. And of course, there are a lot of more clinical trials using our system, but we're not allowed to share any information there. So it's widely used. It's well established in the field and highly reliable. Now, what we would like to do is to scale up a bit more. And for that, we will develop the T-cell transduction large scale. On one glance, so it's planned to be available in summer 2022. And we have actually scaled up or enlarged the cultivation chambers. In our original T-cell transduction process, we were limited to 250 ml culture volume and around about 5 × 10⁹ of T-cells in the end. And now with this new process, we are scaling up to 600 ml and expecting 2 × 10⁻⁻⁻ T-cells in the end. So this will be available soon, hopefully. Then last year, we released our T-cell engineering process, and this process includes the CliniMax electroporator. It's also now an automated and closed way to electroporate your cells. The tubing of the electroporator and the tubing of the Prodigy form one big closed system. The electroporator is controlled via the CliniMax Prodigy, and we can now electroporate up to 157 ml of cells, really large volumes as well. Our process that was released for the electroporator is called the T-cell engineering process, and it looks very similar to our T-cell transduction process, only that now in the gene engineering step, you could choose whether you want to do electroporation and or viral transduction. So you can do both or one of the methods to really go for complex gene engineering. And then what's also really a big advantage with our system is that you can really choose your electroporation parameters. So you can pulse your cells either with one pulse or with two subsequent pulses. And for the pulses, you can choose the voltage, the length, the mode, the polarity, and burst length. So you really have full control over your parameters and can really optimize the parameters for your cell type and your gene engineering technology that you are using. And then last but not least, our customized application service. This is currently the way to integrate your final formulation and final fill onto the platform. So we have the formulation unit released. It comes with a specific tubing set, the CliniMax formulation set, that already includes a waste bag, 4 product bags, and then 4 product or QC bags. We also offer CliniMax formulation and CryoKit. So these are buffers to either have a fresh out or frozen out product in the end. And you can program your process with our customized application service for the formulation unit. And of course, this was just a really quick overview. So if you want to find out a bit more, you can visit our webpage. And we have a really nice webinar from last year about non-viral gene engineering. We also have a nice brochure for our generic or original CAR T-cell manufacturing process. And for each of our applications, we also have application pages that you can visit to find out more. And with that, I would say stay tuned, and you can follow us on different social media channels. So we're quite active there. And usually, if there are news, we will promote them there. And just to summarize this part, we would be very happy if you would choose us as your partner for your cell and gene therapy journey. As Saskia already showed very nicely, is that we can offer research workflows as well as clinical workflows, and we make really the transition quite easy with our products. With that, I would like to close the session. Okay. Now we dive into the analytical assays that, of course, go hand in hand with our workflows to generate engineered immune cells. A short overview what we will be talking about. So first of all, very quick introduction. Then I would like to dive into the analysis of the tumor microenvironment, very important part, of course. Then the phenotyping done via flow cytometry, which is also key in a lot of different assays. Then last but not least, functional assays. This is also a hot topic currently. Good. Then let's get started. So this is something we've already seen in the first part, just to kind of quickly review. So there's a huge pipeline for cancer cell therapy products coming up. So a huge amount of new products being in preclinical phase and also in clinical phases. However, when you think about how many made it already onto the market, it's just 6. Yeah, you can say it's just 6 or already 6 made it. But what it actually says is that there is quite a bottleneck. So there are a lot of projects ongoing, and it is not always a success story. And one of the big hurdles definitely are the analytics. So the analytical assays that go along with IPC-QC monitoring, but also in earlier phases. So as already mentioned, analytics, they are really needed in all different phases. And I think this graph we have also already seen in the first part. So we have different phases that eventually hopefully lead to a commercial product. And we start with a discovery phase with target identification, validation, then lead identification, the preclinical development, hopefully leading to clinical studies, and then hopefully at some point leading to commercial products on the market. And especially in the later phases, in the clinical trials and also in the approval phase, there are quite a few challenges connected to analytical assays. Just to name here a few, costs are always a main challenge. Also, shortening the vein-to-vein time is usually a main challenge. When we think about our analytical assays, also regulatory requirements are putting quite a pressure on. And also from a biological point of view, we are working here with a living drug. So we really need robust assays that can deal with this kind of drug substance. So what should be kept in mind when thinking about your IPC and QC? So you should definitely start thinking about it very early. So when you're thinking about your manufacturing, already think about your IPC and QC because both need to go hand in hand. And definitely the IPC and QC part should not be underestimated. So we really need robust and reliable assays for analyzing this living drug. Also, the regulatory requirements need to be kept in mind. And the costs for the IPC, QC, and also the labor time should definitely not be underestimated because it can really add up. And when we come back to this figure here with the analytical assays in different phases, we can also say that there are different requirements for the assays in the different phases. So what we always want are, of course, reliable assays and fast and easy workflows. But then when we think about flexibility, this is something that we really need in the discovery phase. But then later, when we reach the clinical phases, we don't need the flexibility anymore. We rather want standardization. When we think about the different methods that are being applied in different phases, so of course, there are really a wide array of different assays. So microscopy, for example, qPCR, flow cytometry, ELISA, cell culture, microbiology, just to name a few. It's definitely not a complete list. And we can also not touch all of them now in this section. We will just touch 3 of them. So microscopy, flow cytometry, and cell enrichment for genomic analysis. So starting with microscopy, so what's very important in the research, but then also later again in the immune monitoring is the analysis of the tumor microenvironment. And for that we have 2 microscopic solutions. One is the Maxima. With that we can really characterize TME components and showing their spatial distribution, and we can really analyze over 100 of markers in the same section. And that's really interesting to identify tumor-specific target candidates and then also to see how certain drug products really work, if they are really effective or not. With our ultra microscope, we have different versions of it. We can do section-free 3D histology analysis. Again, we can see how exactly our cell therapies are working in a 3D environment, which is also really interesting for sure. Just to show an example here on the left for the Maxima, so this is a nice poster that you can also find on our webpage. So there they used the Maxima to evaluate target markers in a pancreatic adenocarcinoma. They also used it to analyze the CAR T cell infiltration and then also later if the target marker really remains in the tissue or if there's a downregulation. And also they used the Maxima to see whether the target marker that they wanted then to focus on is also expressed on healthy tissue or not. Then for the ultra microscope, I have here a nice video for you so that you can see it in live. What we can see here is again a pancreatic carcinoma, and in pink are CAR T cells, in green the tumor cells, and in orange the vasculature. So there you can really see in a 3D model how the CAR T cells infiltrated the tumor and how the CAR T cells are interacting with tumor cells, which gives you some more in-depth knowledge about your tumor microenvironment. Then I would like to move to the next topic, which is cell phenotyping done by flow cytometry. And we have our MaxQuant family for that, which can be used in all different phases from research really to immune monitoring. And for the different phases, we have our different models, all of them having different benefits. We also love to support you with our protocols. So for the research and preclinical phase, we have our Miltenyi Biotec tested panels now for a lot of different applications already, CAR T, TIL, Treg, and so on. And you can just check it out on the webpage. And then for the later phases, for IPC and QC immune monitoring, we have our special protocols. And there we have, for example, some for CAR T cell manufacturing, which you can also find on our webpage. If we're thinking about the later phases, IPC and QC and immune monitoring, we would highly recommend our MaxQuant 16 for that, because it really offers the best features and benefits for this phase. When we think about reagents, of course, we have your challenges in mind as well. So what we developed is our reaffinity technology. So we have designed recombinantly generated antibodies, and they have a mutated Fc region to really minimize the background. And they all have one universal isotype, so make the isotype controls easier. And for the research and preclinical phases, we would highly recommend our reaffinity single antibodies, because they give you full flexibility in your panel design, and you can combine them with your CAR detection reagent. For your clinical phase, we would highly recommend our StainExpress dry antibody cocktails and also our CAR detection reagents. So for those of you who don't know our StainExpress dry antibody cocktails yet and don't know our CAR detection reagents yet, let's dive a bit more into detail into them. So our StainExpress, those are dry antibody cocktails. So you have one tube with all the antibodies in there already, and you just need to add your sample. They are really easily stored at room temperature for 1 year, and you can flexibly add your CAR detection reagent, your reagent of choice, to the panel. And we have already 2 panels available there, and of course we are working on more. On the other hand, we have our CAR detection reagents, and we have already quite a few available there, targeted against CD19, BCMA, CD22, and CD33. And for CD19 and BCMA, we know that they can detect all the commercial CAR T therapies. And just for your interest, we have 2 ways how we detect the CAR. One is antigen-based, so part of the antigen is included in the CAR detection reagent. And we also have another method, which is an anti-idiotype detection method, which is then very CAR specific. And this is some data of our CAR detection reagents. So we tested them on CAR T cells, which you can see on the left side. So we tested there our CD19 CAR detection reagents, our BCMA, CD22, and CD33. And of course, they are not cell type specific, so you can use them on any kind of CAR cells. On the right side, for example, you can see CAR NK cells, where we tested the CD19 and the CD33. And as mentioned before, we really wanted to minimize unspecific background, and especially in the CAR detection reagents, that makes completely sense. For example, for patient monitoring, when you have really a rare CAR T cell population in the blood, you really want to reduce your background. And what you can see here on the left is a healthy donor. So this person never got any CAR T cells. And you see in panel A, no CAR detection reagent was added, so very low signal. On the right side, you see our BCMA CAR detection reagent added, still very low signal. And then on the lower panels, you see CAR detection reagents from 2 other vendors, and there's quite some high background. So this is something really to keep in mind, that you should work with reagents that have really a low background signal and don't give you false positive results. Also, we would love to support you with our software to really standardize and automate your workflows. So we have there, for example, our express modes. They are doing fully automated your gating and analysis for you. So what we have done there is that we have analyzed a lot of different samples, up to 1,000 samples, and built an algorithm that then does the gating completely for you. So not only the gating strategy, but really decides where to discriminate between populations. So there that you can really reduce human biased decisions. We also have our Smart Gain technology, which is our software to really have comparable results from different devices and maybe different manufacturing sites, so that you have really a multi-instrument alignment over maybe different continents. What we also offer is a software add-on, which is called 21 CFR Part 11 compliant software. And that includes then an audit trail so that you can always check and document what has been done. And it also includes a user management. To sum this up a bit, so what we would recommend is a complete package for your IPC and QC and also immune monitoring to go for a MaxQuant 16 system, use the CAR detection reagents together with the StainExpress cocktails, and let the analysis be done via our express modes. And we already have 12 panels available for CAR T-cell processes, and we are also working on panels for the CAR NK manufacturing. And this is an example data set. So this is cells stained with our CAR detection reagents and StainExpress and gated and analyzed with our express modes. So what it does in this panel, it excludes the debris, it gates on the leukocytes, excludes then the dead cells, gates on the T cells, and then checks for CAR frequency, and then still checks how many of the T cells that have the CAR are CD4 or CD8 positive, and also looks at that ratio for the CAR negative cells. And as we know that monocytes are quite a challenge in our manufacturing, we also have a monocyte contamination gating there. So this was just an example of one of the 12 panels.
Thank you, Melanie, and hi, everyone. All right, so next I would like to present our solution for magnetic enrichment of CAR-expressing cells. As indicated on this bar on the left, this kit is especially relevant for the preclinical research and also the immune monitoring phase. So the Max Prep CD19 CAR microbead kit can be used for the isolation of CD19 CAR T cells from whole blood. And this is especially useful for downstream flow cytometric analysis with increased sensitivity, for in vitro functional assays, for example, co-cultures with target cells, or genomic analysis, such as gene expression analysis. The kit is compatible with the commercial CD19 CAR T products on the market, and the protocol, unlike other conventional protocols, does not require density gradient centrifugation. The kit includes buffers for erythrocyte aggregation and sedimentation, and the staining reagents for the subsequent labeling of the CD19 CAR engineered cells. So first, the CD19 CAR is recognized by the CD19 CAR detection reagent biotin, with which you are already familiar now. And then magnetic antibiotin microbeads are employed to allow for cell separation. Our microbeads are nano-sized and the smallest beads on the market, actually. So in combination with a high magnetic gradient in our max columns, this technique requires minimal labeling and is compatible with all downstream applications. So to achieve CD19 CAR T cell isolation, the column is placed in the magnetic field of a max separator, as you can see here. And by running the cell suspension through the column, the CD19 CAR cells are retained, while other cells can be washed away easily. So then the CD19 CAR cell fraction is eluted by removing the magnet. There's also an option to automate the separation process with our AutoMark cell separator. Here you can see an example of CD19 CAR cell enrichment. So in the upper right quadrant here, you can see the CAR positive viable T cells. And before enrichment, they constituted 4.21% of cells. And after the separation, the purity of the suspension is increased up to 96.67%. So this shows that a high purity can be achieved of CD19 CAR cells by using this enrichment kit. With longer time periods since the infusion of the CAR T cells has taken place, the blood can really have very low frequencies of residual CAR cells, as is shown in this example here, where it is only 0.27%. So in this case, after separation, the percentage was increased up to almost 80%, and this can be even further increased if needed by using a second column. So even with a low frequency starting material, a high purity of CD19 CAR cells can be achieved. Next, I would like to talk about functional profiling of cells, which is a major challenge during quality control. But nevertheless, it is really indispensable to obtain reliable and reproducible functional analysis, because it's crucial to predict treatment efficacy. Okay, so let's see how we suggest to do that with our solutions in the next slides. So there are many different ways to investigate functionality of CAR T cells, and one of them is to set up co-culture assays with antigen expressing and antigen negative target cells. So by encountering an antigen expressing target cells, the CAR T cells will respond by proliferation, the secretion of pro-inflammatory soluble factors such as interferon gamma, interleukin 2, TNF alpha, perforin, or granzyme B, for example. And this ultimately can lead to target cell lysis. And so, as you notice, there are many different readouts for functional assays. And the one I would like to focus on today is the cytokine response. And I want to introduce two of our solutions for flow cytometry, which enable automated acquisition and data analysis with our MaxQuant analyzers. So the first detection assay is the MaxPlex cytokine assay, which, as displayed on the bar on the left, is most relevant for research and preclinical assays, as well as IPC-QC during manufacturing. So this kit allows you to analyze up to 12 secreted cytokines in parallel. It's a multiplex bead-based assay, which is compatible with standard flow cytometers. It is truly quantitative, so standards of known cytokine quantities are provided with the kit. And if you don't want to analyze 12, but let's say 3 cytokines of interest, you can mix and match a smaller individual panel. Yes, so we have also solutions for human and mouse samples, and we have the express modes to automate your data analysis, enabling unbiased gating decisions. So let's take a brief look on how this assay works. So as you can see here, the beads are coupled to anti-cytokine antibodies, and the cytokines are bound by these specific antibodies, and then they are labeled with APC conjugate antibodies for quantification. Okay, let's take a look at an example experiment. Here, the supernatant from co-culture experiments of CAR T cells and target cells was analyzed using the MaxPlex kit. And as you can see in the mock control and in the cultures with a non-functional CAR, no or very little cytokine release was detected, whereas high amounts of interferon gamma, IL-2, TNF alpha, and GM-CSF were observed upon CAR T cell activation. So this data, which is already published, you can also find in this application note on the right for more details. The second possibility to assess the cytokine response is the rapid cytokine inspector kit. So similar to the MaxPlex kit, the cytokine assay is also most relevant in the research and preclinical phase, as well as IPC-QC during manufacturing. So this kit allows you a combined surface marker and intracellular cytokine staining in one experiment. It's a fast and easy solution for activated T cells. It contains an antibody cocktail, so no tedious pipetting of single antibodies, which could introduce errors and variation is needed. It also comes with Brofellin A and all the reagents for the fixation and permeabilization of cells. And then you can just choose your anti-cytokine antibody of interest, which has been optimized for usage in combination with this kit. We have the solutions for human samples and, of course, the express mode for the automated analysis. As you can see here, through the addition of Brofellin A, the cytokines cannot be secreted and remain within the cells, and this is why they can be stained intracellularly. You can choose between a CD4, CD8 T cell detection cocktail or CD4 or CD8 single solutions. So here again, some example data. You can see the staining of interferon gamma, TNF alpha, and IL-2 in the PE channel. And you can see that the amount of cytokines inside of the cells is increased in co-cultures with antigen-expressing target cells compared to the ones with antigen-negative target cells. And of course, a CAR detection reagent can also be added in the panels in one of the 3 channels. To sum up, here you can see again the introduction slide with the requirements throughout the CGT drug development process. And as you can see and probably know from your own experience, some needs stay the same and some change as you progress in the clinical trial. So you will always need reliable assays and fast and easy workflows. Whereas in the early discovery phase, you will need flexible assays to answer different scientific questions. But as you progress in the clinical trial phase, highly standardized assays are absolutely required. And the products we have just presented to you are designed to fit exactly these needs. So we have the right imaging tools for target discovery, as well as our reaffinity recombinant antibodies, which you can use to design your individual highly reliable panels, as well as our ready-to-use kits and antibody cocktails. And for the highest standardization needs in flow cytometry, the full workflow package with the MaxQuant and the express modes, our StainExpress dry antibody cocktails, and the CAR detection reagents. And with that, I would like to thank you for your attention and participation in this webinar. And if you are interested to learn more about our solutions on our CGT Analytics webpage, you will find some more detailed information and also a lot of products that were not presented today due to the time limitations. We also have compiled some interesting material for you about the tumor microenvironment, our CAR detection reagents, and the express modes. Feel free to reach out to your local sales organization, and thanks again for your attention.
Thank you again, Drs. Graband, Rietenbach, and Rösch, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care everyone. Goodbye.